A good model to go over the some of the female structures. That structure, very important structure, is the ovary. And one thing you'll note if you look at the ovaries now, like this ovary over here, even that, they're actually trying to show follicles. And so when you are looking at a chart showing ovaries, you'll often see a corpus luteum or a mature follicle. I probably won't ask it like on the chart. I will use our nice models for those follicles. But when you're looking at ovaries now, you'll see things like, you know, this little dot right here, this little dot right there. I don't know what they're trying to show, but it is a follicle or remnant of a follicle they're trying to show. And so whenever you see a follicle, you can try to identify it, but I won't ask it the follicles unless it's some of the models we've already kind of gone over. In the microscope we have follicles, and then this model and that other model are the main ones that ask the follicles. And so there is the ovary. This structure there is the uterus. Entire structure is the uterus, and then we're going to be looking at this structure next, which is the fallopian tube or the uterine tube or the oviduct, and you can use either term. And so what we're going to do is look at a cross section through the uterine tube. So what you would expect is a tubular structure, kind of like what we've seen with other tubular structures. The digestive system, we saw a lot of the tubular structures. So this is the uterine tube, and I think most of your slides will look something like this. And so you can imagine the whole uterine tube is there, so we could move it around and we could see the wall, and then this white space would be the lumen. And so you have the lumen, you have this muscular layer, you could probably guess, even without looking, is muscularis. Just like we've seen muscularis in many places, a lot of these smooth muscle layers of these tubes is called muscularis. And so we then go into the lumen, and the membrane type, or the layer lining the lumen, would be the mucosa, just like you've seen in other places. You have the mucosa, you have the muscularis, and then the outer layer would naturally be called the serosa. If you see a nice smooth layer where it looks like a serosa. And so you have serosa on the outside, mucosa on the inside, and then you have a muscular layer called muscularis. That should be pretty familiar. One thing that's a little unique about the uterine tube here is you can see there's kind of folds of mucosa filling up the lumen. And so that's what we can see all along here. And the tissue type of these, the mucosa here, there's two types of tissue. And so that's what we'll look at. And in your lab manual, there's only one circle, but you definitely want to be ready for both powers. This is just low power at 40 times what I'm showing there. And then this is high power. And so I probably won't require that you draw this picture. However, I kind of like this one better than this one, or they're both very good. And so then it comes, I probably like this one better, but it comes also sometimes down to what the other instructors want. And so either one of these could be on the exam. The beauty of this one is we can see the tissue type. And so what I'm going to do here is, this is low power, so it's easy to see mucosa, lumen, muscularis, etc. To really see the tissue of the mucosa, we have to go to high power. And so what I'm going to do is just go to high power at one of these little pieces of the mucosa, and then it will look like this. And so now I'm looking at high power at the mucosa, and when I look closely, I will be able to see cilia right there. And it's much easier in the microscope to really see it, just zooming in a little more, but you can probably see right here in this picture, this is cilia, just like you saw in the trachea. And so you may remember cilia in the trachea, and here we have cilia in the uterine tube. And so the function of this cilia is to move that secondary oocyte, or the fertilized ovum, from the, to the uterus, basically. So the cilia is going to be having little waves to move that secondary oocyte. This is the video inside of the uterine tube, and you can see the cilia moving. You can see the sperm are trying to fight against the cilia, but there's one place here where you can really see the wave, right here. You can see those cilia just moving in the waves of cilia, and the cilia are trying to move the secondary oocyte or the fertilized ovum toward the uterus. Kind of interesting, the sperm are trying to swim the other way. So the sperm are going the opposite direction of where the cilia is trying to take them. But we can see real cilia right there in this video. Then we go back to our slide here, and so these are the cilia that would be moving these wave-like patterns, and they are functioning to move that secondary oocyte or fertilized ovum. So that's one tissue you'd see, and that's what makes it kind of interesting, is you can see that cilia. And one other point with cilia, you may remember with the trachea, smoking does paralyze cilia a little bit. And so it inhibits cilia. And so smoking can cause a chronic cough because the cilia don't work so well and get the mucus up. But also with smoking there's a higher risk of ectopic pregnancy. And that's also why it's good to see the cilia. Does that make sense? That if you paralyze this cilia, now that secondary oocyte might not get to the uterus, and then the pregnancy could begin in the uterine tube, and that would be an ectopic pregnancy. And that's why you'll hear that smoking has a higher risk of ectopic pregnancy. A pregnancy does not happen in the uterus. One other tissue or cell that you'll be looking for here is non-ciliated. And you have to look very closely. Most of this is cilia. But I go to this particular cell right here. This cell does not have cilia on it. And the lab manual calls it non-ciliated simple columnar epithelial tissue. The book calls it a PEG cell. And so you could definitely call it a PEG cell as well. I think I have a higher view, a little higher view right here. So this right here, this cell right here is called a PEG cell. Or you could call it non-ciliated simple columnar epithelial tissue, either one. And so there's cilia there, and then this cell, and those are pretty good examples of PEG cells. I can see a little bit of that apical membrane right there. And so I tried to draw a picture of how you would expect it to be. That nucleus actually comes out closer to the apical surface than the nuclei of the ciliated cell. Here is the nucleus of the ciliated cell. This is the nucleus of a PEG cell. And so that's one way you can see. When you look in the microscope, you should actually see, oh, I can see there's no cilia right there. And so you want to be able to recognize those two types of cells. The PEG cell, or this non-ciliated cell, secretes fluid. And that fluid will help to nourish that secondary oocyte or the fertilized ovum. And so what you want to do then, to be able to recognize the low power here and then the high power here. Just some other notes. Going back to the low power, this is the muscularis. You want to know the function of the muscularis, and that is peristalsis, just like so many muscularis is. Peristalsis, in this case, this muscularis is going to be moving that secondary oocyte or fertilized ovum also toward the uterus. So there's this little peristalsis, very gentle peristalsis. This muscularis is not very thick, so the peristalsis is very gentle. And you'd want a gentle peristalsis because you're moving that secondary oocyte or fertilized ovum. When we look at the vas deferens for the male, it will look very similar, but that muscularis is going to be huge because in that case, you are going to be wanting to eject sperm very quickly. And so that's one thing that I will kind of bring up again is this muscularis is thin here in the uterine tube. One other thing that is worth bringing up is when we look at the lumen here of the uterine tube, you can see all of these folds of mucosa. Can anyone think why the uterine tube would have all of these folds? There is a pretty good reason. And so you can see the lumen is not the other thing, why is it like this rather than just having a muscularis and then having, just a nice lumen inside? Why do we have all those folds? Anyone think? So the reason is because really of the cilia. And so the whole goal here of the uterine tube is to transport that secondary oocyte or the fertilized ovum to the uterus. And so now imagine if you didn't have the folds and it was more like this, the secondary oocyte would be like this, and only one part of that secondary oocyte is touching cilia. And so it's not as effective, but now we go over here and imagine the secondary oocyte being right here. And, or even, I'm kind of made it close to that circle, but now if the secondary oocyte is right here, can you see that this secondary oocyte is being moved by cilia on this side and by cilia over there? And realistically, you can surround the secondary oocyte by these folds, and then you have cilia moving it. And so you have all these folds just so that you can have more cilia helping to move that secondary oocyte. And so in this case, you don't have as much cilia as you would here helping to move that secondary oocyte. And so it's good to know kind of why you would have all those folds. Does that make sense? So that is the uterine tube.